Shall we start?
Okay, Doctor. Yeah.
Good morning to Professor N. Senthil Kumar ji, and good morning to all the participants who have joined. Participants, we are having today Professor N. Senthil Kumar. He is presently Dean of Life Sciences, and earlier he has been head of the Department of Biotechnology, 2 terms. He is having exposure to international universities. He has worked in China for an university for some length of time. If you see his career, he's having experience of more than 20 years, but in this 20 years, it is, I will say, a very short time period. He is having research papers more than 140 in 20 years. He has written more than 3 books, more than 40 invited talks he has given. Presentations of seminar and other oral presentation he has done 25 plus. He has attended many training programs. If we see his publication history, his citations record in last year for Google Scholar, it was 3031. I-10 index is 68. Cumulative impact factor is 250. So, if you see his publication, his research guidance, he has so far produced 15 PhDs and 6 are undergoing, and he has produced 10 MPhils under him, and 2 are undergoing. So he's having a very, very impressive biodata. He's a very good speaker. And sir, we are having some 28 participants in this course induction program. So all are new faculty members. And coincidentally, we don't have any faculty from Mizoram University.
Okay.
All are from other states. Few are from Assam, few are from West Bengal, few are from southern part, few are from central India, from BHU 3. But again, coincidentally, there are 2 faculty members who are locals of Mizoram, who studied here, and now they are employed in Banaras Hindu University.
Okay. Okay.
So we are having a good mix of what to say, faculty members from different subjects, from different parts of India.
Right.
So with this short note, I would like to request you to start your presentation. Sir.
Yes. So thank you, Professor Bhartendu, for the good introduction as well as for this opportunity. And of course, our UGC HRDC Center of Mizoram University has been always, very proactive and conducting many programs continuously throughout the year. So, and it is always very good opportunity for, for us, for you and me also to meet participants throughout the country each and every time and interact with them. So it's basically an exchange of knowledge. And like you mentioned, it is a collection of different disciplines or subject teachers. So from various universities across the country. So I will also keep my talk as basic as possible. And I would always be very happy to interact and answer questions or clarifications or hear from the participants some new ideas or suggestions. So once again, thank you, Professor Bhartendu, and I'll go into sharing my screen. So I'll switch off my cameras and I'll start sharing the screen. So I hope
yes,
the screen
is yes, full screen is.
Okay, thank you, Doctor. My today's topic, I have kept it as human diseases, biological and cultural aspects. I'm going to talk about the risk factors for human diseases from the biological point of view, as well as from the cultural aspects. So how does the human disease, right? Disease themselves evolve, and how is it manifesting in the populations? We are also going to see why it is different from different populations, the disease patterns, the nature of the disease or the type of the disease, and what type of interactions happens between the human system, or basically the genes and the environmental factors. So as such, we are going to discuss about the components of various risk factors. This is one of popular proverb. I mean, every evolutionary process leaves their distinct mark and trail on their genetic material. So, like if anyone asks us that, how old are you, right? So, biologically, the answer should be, I am 2 lakh years old. So, approximately 2 lakh years before the Homo sapiens, we are still Homo sapiens, right? So Homo sapiens appeared on this earth. And of course, there has been a lot of changes at the cultural level, at the environmental level, at the biological level. And as we all know that, at every stage, we acquire knowledge, we adapt to the environment, we adapt to the stressful conditions, and that information and knowledge is stored in the genes, and it is being transmitted to the next generations. So this can be one of the reasons why the IQ levels in younger generations are much higher than the previous generations. Because when we transmit our genes to our children or our offsprings, which has been happening for maybe the last, like we said, approximately 2 lakh years, each and every time the adaptations for surviving better in this world, survival strategies are being stored in the genes and passed on to the next generations. So we all know the evolution of mankind also a little bit, like from a foraging society, from a nomadic life. We moved into a pastoral society, then into horticultural society, then agricultural revolution came into the society. Right? So then started, once mankind stayed in a place and then started doing agriculture and evolving, then came industrial developments, and now we are in an era of technology development, or it's a techno society basically. Knowledge, right? It's a bit, otherwise we call it as a knowledge society. So with this basic introduction, let me move on. So as we see here, the diseases, when we talk about human diseases, generally it can be of two types broadly. There can be many other types of diseases, but in a broader sense, it can be monogenic or complex, means multigenic. So in a monogenic disease or disorder, it involves a single gene. Like as humans, we have nearly about 25,000 genes, less than 25,000 genes. And each gene has their own functions. Each gene has their own, structural sequences, their functions, and their evolutionary rates and patterns are different from each other. So if a disease is linked to only a single gene, one classical example is cystic fibrosis. So that disease is called as monogenic, single gene disorder. So mutation in a single gene, it follows what we call as a Mendelian pattern of inheritance. So any mutation or changes in the ATGC arrangement of a single gene can lead to the disease. It is sufficient to cause the disease. And this, can be only change that what this gene can bring in among different individuals is either it can be the early onset or late onset of the disease, or it can be slightly varying among the different male and female individuals. So in a family also, if we see the inheritance pattern, it can be dominant or a recessive pattern of inheritance. How many individuals are affected by this disease determines whether it is a dominant pattern of inheritance or recessive. In a family of a male and a female having 3 offsprings, as we see here. So this is a pedigree chart. What generally we plot for any type of human disease patterns. So the impact of mutations on the single gene affecting the disease phenotype can be 100%. And the genetic risk in the population also is going to remain uniform in different families because it's a single gene. So mutations in the single gene will lead to the disease, and it is common among different families. So it follows a simple pattern of the inheritance and the etiology, whatever we call. So in a complex disease, it involves many genes, as we see here. As I said, there are less than 25,000 genes. For example, cancers, right? If we talk about breast cancer, at least 50 or 60 genes have now been implicated, which can involve in the process of breast carcinogenesis. Similarly, stomach cancer, at least 70, 80 genes can be easily attributed. Based on the literature and based on the studies in different populations, these findings are already published and it is well accepted as well. Even in the case of diabetes also, there are a couple of genes that are being involved. So that we call it as a complex disease if there are involvement of many genes at the biological level, right? So now we are talking about the biological aspects, whether it is a single gene or a double gene or multiple genes. Here, because there is involvement of multiple genes, so no single gene is directly responsible for the disease. It is a collective involvement, and at the same time, it also needs some environmental influence. Like when we say environmental influence, lifestyle factors and food habits, etc., can influence this type of diseases. So the gene variants or mutations, basically when we say the gene sequence, it is composed of only 4 nucleotides, ATGC, right? Adenine, guanine, cytosine, thymine. It makes the 3 billion base pairs that human genome consists of. So within that 3 billion base pairs, less than 25,000 genes are present. So here, there is a lot of interaction between gene to gene, gene to environment, and environment to environment as well. So that's why it's called as complex disease, right? So, and finding out a cure for this type of complex diseases are also complex, right? Because it's not a single target that we need to attribute, but it needs multiple targets to tackle. And at the same time, because this is kind of an synergistic interaction that happens, so we cannot just regulate the disease pattern just by studying few factors alone. So similarly, if we draw a pedigree chart, right? So cancers run in families, right? We all know that. There are familial cancers or hereditary cancers. There can be cancers, that are acquired during a lifetime of an individual. There can be cancers that are cancer genes getting mutated that can be just inherited from the parents. Even when a child is born, we get 1 copy of the genome from father, like let's say 1 allele copies of all the genes, and 1 copy from the mother. So if the parental genotypes are mutated, which can lead to any type of disease, then the child obviously inherits it, and that becomes germline mutations, right? Or if an individual doesn't have any germline mutations at the time of birth, but during the adulthood or because of smoking or drinking or other types of environmental risk exposures, if the individual acquires a mutation in the gene, then we call it as somatic mutations, right? So if we see the inheritance pattern here, this is also complex, right? So some individuals in a family of this pedigree chart, like we see, for example, can have the disease. Let's say the darkened circles are the diseased individuals, and the undarkened circles or squares are the healthy individuals within a family. So here about 3 generations have been plotted. So the impact of variations in different genes, right, is only about 30%, right? So when you calculate the genetic risk in a population of A, B, C, D, different populations, then the impact of the variations in different genes or for leading to the disease is only about 30%. And the remaining rest is taken care of by the environmental risk factors or the lifestyle risk factors or food habits, etc., etc. So obviously what happens is, when you compare across different families also, different individuals, right, have varying levels of risk for the same disease. So in short, if I have to say, or if I have to explain it with an example, let's say, take the example of breast cancer, and the 2 important breast cancer genes are BRCA1 and BRCA2. So in short, it is called as BRCA1, BRCA2. So these are the 2 genes that are primarily involved in the mutations in these genes are primarily involved in the initiation of breast cancer. So necessarily the mutations, the percentage or the degree or the number of mutations that need to happen in a particular population for the breast cancer need not necessarily be the same across the different populations. So going back to the monogenic disorder, like it's a single gene, example cystic fibrosis. The name of the gene is CFTR, and it is said that it can have up to 1,500 variants or mutations. So if variants are there, disease will be there. The only variation that can happen with this one particular cystic fibrosis disease is the severity or the age at onset may vary among different individuals, right? But the disease is for sure. The phenotype or the disease condition is for sure. And the risk is the same in all the families. If there is a gene mutation, then there is the disease. So it's the same risk. But if you look at the complex diseases, the risk of developing a disease can range from low to high. Right? As we can see, different lifestyles, different environments, different gene variants, different genes involved. When you list 100 genes for a particular disease, necessarily all the 100 genes need not be mutated in a single cancer patient also, right? Only 20 genes may be mutated, right? Or gene mutations might be there, but it may not lead to the disease unless and until exposed to a particular environmental or lifestyle factor. So multiple genes and variants, when we say variants, it is basically the mutations. The varied risk factors are present. There is no particular inheritance pattern, and there is always a mix of genes and environment that have to act together. So this is a summary of what we were discussing in this slide. I can stop here if anyone wants me to explain something, or if there are any clarifications or questions.
Sir, participants are not having access to audio, so they cannot put questions, but they can put the question in chat box. So far, no question. So participants, if you are having any question, please put in Q&A box.
Yes,
so that sir may respond to it.
Yes, because this one single slide is a bit complex.
Yeah, many things, many facts are there.
Yes. So if the participants want any clarification, we can solve it and then go ahead.
I think, sir, better you continue. If anything is coming, it will come in chat box.
Okay, sure.
And participants, don't wait till end. As soon as any question is coming to your mind, just put it. Yes. Ventilate it into the chat box.
Yes.
I know Professor Senthil Kumar, he likes more discussion than one-way presentation. So he will be welcoming it.
Yeah, exactly. Yeah, so it is always good to hear from different people as well. There is something in the chat box. Okay, fine. Yeah, they can put it in the Q&A box as well.
Yeah. Either Q&A or chat box, wherever they feel like.
Yes, sure. So now we go back to little bit of basics. Like we all know the common biological function. We have the DNA or the genes in the cells. The DNA is double structured helical type molecule, and in the cell, it is processed into proteins, right? I mean, or otherwise, the DNA transcribes into RNA, and RNA translates into proteins, right? So for any particular genetic character, right, the DNA or the genes are responsible basically, but they don't act directly, right? They act through the proteins. So proteins are the soldiers. Whether we are, having a black hair or a white hair, of course, the information is stored from in the genes. The commands come from the genes, but the proteins are the ones which actually brings about that particular phenotypes or the changes. So there are a lot of research and studies that happens now with these three types of biological molecules. The basics of life life processes is the informations are stored in the DNA. As we saw, the 25,000 genes regulate our whole system, whole functions, and any mutations or variants present there or alterations present there can downstream affect the RNA and downstream affect the proteins as well. So if you sequence or study the DNA of a healthy individual versus the DNA of let's say, a cancer patient, you will find a lot of alterations, right? About 50 genes or 100 genes will be altered at the sequence level. And obviously, because there is a collinearity between the gene and the proteins or polypeptides, there will be structural changes like we see here. The structure of the proteins are also very, very crucial in regulating the function of the protein itself. So if the structure changes, then the function changes. So that is why people say proteins are very dynamic. They keep on changing, they keep on altering. And the DNA is very stable, basically. Right? They don't want to change as much because then it will change the whole system or the whole body or the whole biochemical and metabolic processes. So that is where the study of these are gaining more important in terms of human diseases as well from the biology point of view. Even the intermediate RNA is also very important. At a given point of time in a cell, thousands of different types of RNA molecules can be synthesized, which necessarily need not convert itself into proteins. It can degrade as a RNA itself. I'll give you one small example. DNA, yes, we all are humans, right? Homo sapiens, we belong to the same species. So our DNA has to remain the same. At least 99.9%, the DNA sequence remains the same, whether we are an African or an American or an Asian or a European. But the 0.1% variation in the gene sequence can also bring in a lot of changes. That's a different story. So even though the DNA is 99.9% exactly the same in all the people, here one example is like imagine all the 26 of us who are now currently online. We are traveling in a bus, right? And imagine the bus meets with a small accident. So what happens to the 26 of us? It's a small accident. It's not a major accident where all people die, but a minor accident. There will be 26 different reactions, right? From the 26 individuals. Some people would have fainted, some people would be shivering, some people would be, sweating, some people would remain as if nothing has happened, right? Even though we are all humans, our genes are exactly 99.9% the same. There is no change, but the reactions are different. It is because the type of RNA molecules that our DNA produces in response to that minor accident that can differ. And obviously, the proteins can also. So it's very, very interesting to study these type of changes between a healthy individual and a patient for finding out a good, sustainable drug discovery process or strategy. So again, this is one another basic slide. Till now, I was talking about mutation and alterations. So for some of you who are not clear, I'm sure this slide will make you clear that what type of mutations or alterations or variants we are talking about. So imagine this is a, okay, we go much down the lane first. So imagine this is the cell, right? And there is a nucleus in it. Within the nucleus, there are 23 pairs of chromosomes. So each of this pair, one we get from father and one we get from mother. So this chromosome has some strands, right? The DNA strands, like we see here, it enlarges. This image is enlarged here for our understanding. And these are the 4 basic nucleotides, ATGC, right? That we are talking about. Basically, these are the genetic material, right? That we inherit from our parents. So this ATGC arrangement is what is shown here. So over a evolutionary time scale period, imagine there is one individual, and of course the purpose of life is to reproduce. Right? Biologically, if you have to define what is the purpose of life, it's to reproduce, because every organism in this world wants to perpetuate its own species and conquer the world. Be it a small bacteria, right? Given a chance, it will keep on dividing. Right? Every 20 minutes, it keeps on dividing and it makes its own progenies non-stop. So even a plant want to reproduce, even an animal want to reproduce, even humans, we want to reproduce, right? So the basic component, right? The purpose of life is that. And see, imagine this individual reproduces over time and has 2 offsprings. So one offspring has the same arrangement of this ATGC or TGA, GCTA, TGA, GCTA. So it's matching with the wild type, right? Another individual or the second offspring has one mutation or has one variant from G to A. So over time, this individual also reproduces. Imagine it has 2 offsprings. Again, one offspring is exactly the parental type, whereas the other offspring is having a second variant, T to G. And over time, this keeps on accumulating, right? These variations and mutations alterations keep on happening. And so from one ancestral lineage, right, there are a group of individuals, and the second lineage from the same ancestor has another set of group of individuals, which are now slightly genetically different from each other, right? The sequence arrangements. There is variants that are differing from this lineage with the variants that are present in the offsprings of the second lineage, right? Imagine these are the current offsprings, okay, the present day generation. So what happens is at one point of time, even though the ancestral type is the same, but the modern times, due to evolutionary events and changes and the mutations or variants, genetically, these 2 lineages, the individuals belonging to these 2 lineages are different from each other, and they are no longer able to reproduce among themselves. Right? Individuals belonging to this lineage is not able to reproduce with individuals belonging to this lineage. Then a new species has been created. So in humans, in general, if you see, this accumulation of these variants is not so fast. It's a very, very slow process. That's why we still remain as Homo sapiens. Whereas if you take the case of lower prokaryotes like bacteria, primitive organisms, their rate of spontaneous mutations are very high. They can acquire this kind of mutations or variants very rapidly. because their reproductive time is only 20 minutes, right? For us, it is 10 months. So within 20 minutes, it can acquire variants, and the next 20 minutes variants, right? So even if you see the case of COVID virus also, right? Within 14 days, it can acquire one variant, which can even change its nature, right? The from Delta to Omicron, we have seen so many different variants, right? So the lower organisms like bacteria or virus undergoes this type of variations very rapidly, and that is why you can find even 1,000 different species of bacteria or many different types of viruses. And if you go a little higher in the scale, like lower eukaryotes, like butterflies, for example, there are about 500, 600 species. But if you go up higher in the higher eukaryotic level, then it like humans, right? The rate of accumulation of these variants and mutations are very less. So talking about humans and human evolutions, human evolutionary rates, scientists now popularly believe migration out of Africa or otherwise out of Africa hypothesis. And by doing a molecular dating of their genome sequence, it has been found that the age of the African human genomes are much higher than what what we found in the North America or South America, and it is popularly believed that humans appeared on African continent first and then migrated to different parts of the world and populated the entire world. Now we have an excellent roadmap of human migration and evolution, and how based on the anthropological evidences, historical evidences, and genetic or DNA-based evidences, this part of the world is now the old world, and this part of the world is new world now, based on the human. So here are the major, human races, major types. Of course, there are many more human races, but in general, right, the diversity, we capture them here. Even 0.1% of the variation has led to so many changes in the phenotype. That is because of our environmental variations, right? The climate, the geographical boundaries or the geographical regions, the culture, the food habits, the lifestyle habits. Right? It's because of all these changes that we have such a diversity in the phenotypes of the human races. So humans and chimpanzees, right? Chimps are our closest relatives, about 96% identical in the genotype. And as we discussed, we humans are 99.9% the same. When I say 99.9%, it is the arrangement of the DNA sequence, basically. So studies have shown that individual cultural variants, right, within and between the different ethno-linguistic groups, right, and a large proportion of cultural transmissions occur within the group, right, from parents to children's, and from mothers' cultures to descendant cultures. So there have been, not only transmission of DNA or genetic material from parents to their children's, but also along with the genetic material, we also transfer the cultural knowledge, right? Cultural inheritance also happens. So this is where there is a parallel evolution of both genes and culture with respect to the environment. Environment always have a strong influence, right? Um, we always study in biology that the genes interact with the environment to produce a particular phenotype. So with this, we have now a strong understanding of how genes and culture evolve, or how biological evolution happens, or how it interacts with the cultural evolution. Which is first? Culture is first, or genes are first, right? It's a tough question to answer. It's just like answering whether the chickens came first or egg came first, right, in this world. But there is a strong interaction and correlation uh, between both. So we always believe in natural selection. Nature selects best of the best genes only to reproduce, only to survive, only to interact, right? And it allows only genes that are strong enough to survive in this world to reproduce further. So that is one of the reasons why these days we have so much of infertility problem as well among human races also. This is both in the Western world or in the developed nations or in the developing nations or even in the underdeveloped nations. And this natural selection process tells us many secrets about life. It helps us to even understand life. Not only the genes, the proteins or the RNA help us to understand life, but also the genes and the environmental interactions also can influence. So the evolutionary and the population history are responsible for the biological diversity, right? What we see today. Life is very diverse, right? If you take a plant species, you will find many species within that. If you see a bacteria, there are many species within that. If you see animals, there are again many species within that. So the diversity of the life, right, or the biological diversity is based upon the evolutionary patterns, the environmental influences, and the population history, which also includes the culture and the cultural aspects. So humans, like we saw, were all Africans, maybe the out of Africa hypothesis. And due to climatic changes and availability or non-availability of food, we always go in search of food and water, right? So people started migrating to new environments and adapting to the new environments. And there came, gene evolution also, changes in the DNA and the adaptations. So we know that the African people, because of direct exposure to the sunlight, they are dark skinned, but they are very rich in vitamin D. Whereas the Europeans or the white people, or to some extent the Asian races also, all have vitamin D deficiency because of the white skin or yellow skin, right? And less exposure to sunlight. The skin color is due to the less exposure to sunlight. Obviously, the disease is vitamin D deficiency. Symptoms and syndromes play a major role. So, this is a simple classical example of how environment can cause a disease in humans with the regulation of the genes and proteins. There are certain genes, VDR genes. The gene name is VDR, vitamin D receptor genes in humans, which regulates the level of vitamin D quantity in this body, and that is regulated by the exposure to the sunlight. It's a very, very intricate interaction. So if we broadly see what are the biological factors that can influence the disease and what are the environmental factors. So biological factors, directly we can talk about genetic variance. Variance means mutations, okay, alterations in the ATGC. Variance in specific genes that produce proteins. And we can also talk about ancestry. because ancestry is very, very important to understand the genetic alterations also. And the biological factors, for example, one is skin color. There are multiple examples. Some of the examples we'll be looking into in the course of this talk. And the environmental factors, other than genes, we have so many other factors that can uh, come under biology also. Like for example, diet, food habits, physical activity, or lifestyle habits, exposure to pollution or smoke, right? Or how a toxin can alter the changes in the biology of the system. So cultural factors can also come under environmental factors, and it includes a whole lot of factors like it can be psychological, psychosocial factors, it can be socioeconomic status, it can be stress, right? Whether a person is single or married can also be a stress factor in the society, right? The depression, the happiness, the friendships that we have, the societal inputs that we get on a daily, day-to-day basis. If there is any physical trauma or other type of abuses that has happened to a particular individual, or even the level of education or the level of income, all this can influence the disease and the disease patterns. So before we go a little more detail into the biological evolution, is there any questions? Yeah, people can feel free, I suppose. Okay, there is one good question. How genetic material, or let's say, for example, DNA, are responsible for mental disorder like autism or Rett syndrome or even schizophrenia or any type of mental sickness? It's a very good question. Autistic related genes, for example, are there, which are inherited from the parents. So the parents might be having mutations in certain genes that are involved with autism disease in a recessive condition. But when it is transmitted to the offspring, it can turn out to be dominant condition. If both the parents were having one copies of that, the offspring can have two copies of it. By mistake or by luck or fluke or by probability, if that two copies from the two parents, right? One copy from each parent, if it gets inherited in the children, then the child gets the disease. So there are so much of studies that have been already shown worldwide in worldwide populations describing about 10 to 20 genes that are involved in the autism disease. Especially if you take autism, the child will look very normal, even up to the age of 7 years or 8 years, or even up to 10 years, the child will be very normal. But the disease phenotype will be exhibited only after 10 years or 8 years. So that is where, the study of these genetic factors are so important, that if there is a susceptibility of autism running in the family, when a child is born, if the genes are studied or sequenced, and if we find any mutations or variants in those genes that can lead to the disease, we can easily treat the child at the very childhood stage itself before the disease onsets. Thereby, the disease can even be avoided or easily, tackled, mitigated. Based on nutritional factors, based on certain medicinal drugs. Okay, there are treatments which are available that can even elevate or remove the disease also, if it is detected before the onset itself. So a lot of literatures are available for this type of mental sickness, mental illness, or autism or Rett syndrome. Gene mutations and variants are involved. Of course, there are many environmental factors. Like I said, food and lifestyle habits can also influence it. So these things can be tackled only if we integrate the knowledge at these three levels: at the biological level, at the environmental level, and at the cultural level. Right. So that is where the my whole purpose of this talk is based on this one question only, basically. I hope I have answered your question, but if not, you can again post the question again. It seems, oh yeah, this has been answered. So, in general, biological evolution, right, it's all based on the survival strategies. Like if you see the modern day elephants, they don't have the husk, right? Why? Because there was no need of these big husks for these elephants, because there are very less natural enemies for them, not like the earlier days. Modern days, a very less number of tigers and lions are there which can attack the elephants. So the elephants thought that no use of wasting the energy in the production of this husk, let's divert it to some other useful resource for other functions of the body rather than wasting the energy, right? So energy and the metabolic resources. So these are all some survival strategies, basically. We all know why giraffes also have a very long neck. Simple answer is being during the time when giraffes evolved on this earth, their food all were on long, big trees. Right? They didn't have herbs and grasses much to eat. So it was trying to reach out to this food, and it evolved into a longer neck. So this is all process of biological evolution. So if we again take the case of COVID, in humans also we saw that certain populations, certain races were susceptible to certain strain of COVID virus more than the other populations across the world. Like in the first cycle of COVID, we saw that Italy and the other countries were more affected. Many people died there. That much effect was not there in India. Whereas in the second wave of COVID, with the different strain, right, we found even India was also affected much. So, and then came, finally third wave or fourth wave. We don't know in which wave we are now presently, but the Omicron strain proved to be less dangerous to humans across the world, right? As the virus kept evolving, humans, we also kept evolving, adapting, and we could easily, fight against the virus now, I should say, right? So the cultural and lifestyle and food habits, right? Why Indians were not affected in the first wave? Maybe because we have a very continuous and long-term exposure to many viral diseases like HIV or even other pathogenic uh, parasites like malaria or many bacterial pathogens. We had so much of infections, and our system, we have been evolving along with these diseases, right? Or disease-causing organisms. So maybe that one point was good enough for us to have a natural defense mechanism against COVIDs. And also, like maybe our lifestyle and food habits in general, we have a lot of spice along with us, which have a lot of medicinal properties. So that could also have been the reason, right, why we could come across and fight over. So basically, the biological evolution is a change in the inheritable composition of a population over time. In the sense, generations after generations. Classical example of this is antibiotic resistance. Like if you give an antibiotic to a person who is dwelling in a metropolitan city, right, for a few generations, their ancestors also lived in the metropolitan city, the antibiotic may not work. Because we have been consuming antibiotics on a regular basis, generation after generation, and the organisms have become resistant to the antibiotics. Within 6 months, bacteria can get resistance to a particular antibiotic by changing its DNA or genotype. Right? So this type of biological evolution uh, can give us a lot of secrets about life processes, to understand life better. So rather, if you give an antibiotic to a person who is not exposed to any antibiotics, who is living in a remote corner inside the deep forest, let's say, or in general, the antibiotics works well in tribal populations. The antibiotics works very well for them, right? They get cured immediately, even with a single dose of antibiotic. No need to take for 3 days or 5 days or 7 days or 10 days to get a cure, even from a common cold. Right? So that is one classical example. The next is pesticide resistance. The farmers these days, face big problems like for tackling the insect pests of agricultural crops. They have to sometimes spray 25 times higher dose than the recommended dose of the pesticides to kill the insect on their crops. So this pesticide resistance is because of 2, 3 genes, the detoxification genes present in the insect to detoxify those pesticides, and then they survive much better. So, like our cultural habit was organic farming, which were very eco-friendly and human-friendly also. It did not affect the ecosystem. It did not cause resistance to insects. It did not lead to resistance by the insects. They were, farming in a harmonious way, but by use of chemicals or pesticides, we have not only polluted the environment, but also unable to control the insect pests now. So from organic farming to inorganic farming, let's say, we are changing the evolution of the insects, right? And it is not only insects, it is also the humans, because we eat now rice and vegetables or even drink water with full of pesticides. Right? We even eat fish with a lot of formaldehyde, right? I mean, formaldehyde is a preservative. So these factors, right? The cultural habit, as the culture changes from organic farming to use of chemicals and fertilizers, genes also evolve in a different way. Not like the original ancestral way of organic farming, how their genes were evolving, the humans. Now our genes are evolving in a different way because of change of agricultural practices. Yeah. So these processes of biological evolution can be because of mutations, or otherwise we call it as natural variations. And there is always a struggle for existence, right? Every day we need to struggle for our own survival. Otherwise we'll be lost. We'll be overtaken by someone else. And if we don't struggle for our existence, if we don't perform, so nature will not select us. So the characteristics better suited to the environments always are preferred by the environment itself. So there is a natural selection. Migration, yes, new dominant genotypes emerge in different environments due to changes in either agricultural practices or changes in the environmental conditions, or changes in the geographical boundaries, right? Or changes in the presence or absence of natural enemies. So all this are leading to the processes of biological evolution. So again, when we talk about evolution, like sources of variations are very important. Here in the picture, you are seeing Jane Goodall, right, who was working with chimpanzees for more than 25 years in the deep forest, right? So she studied about chimps and how chimps could use, stones for making fire also, right? Or how they can basically use tools for their own survival and things like that. So the sources of variation are very important. Like, if two brothers from the same house, let's say, are exactly looking the same, it's going to be very difficult, right? Even for the parents and even for the two sons, for example, to survive in this world. Right? So there will be a lot of complications. And imagine if all the human individuals are exactly looking the same. Right? Like you enter into a forest of teak cultivation, and you will see some 1 lakh trees exactly of teak plantations, like a monoculture. You don't find any diversity there. You don't find any variation there, right? And it's not good for the environment also. There will be a lot of ecological imbalances. But if you go into a natural forest, you will find 1,000 different varieties of plants. So that balances the environment in terms of food requirement or nutrient requirement or other pests, insects, and things like that. So the source of variation, right, is sexual reproduction. Um, like I said, one copy we get from father, one copy from mother. Each of these copies, they combine again and interact again, right, in our system. So that is where, new combinations, probabilities arise. And that is how even if there are 10 brothers in one family or one house, they all look different. And there is always a method of selection, right? So nature always selects the best of the best genes only to reproduce and which can compete and which can survive in this world. So individuals who carry weak genes are not allowed to reproduce by nature. But nowadays we have IVF and so many other methods by which we still reproduce. But in general, this is a natural process basically. So the third mechanism is a mechanism to retain the changes and transmit to the younger generations. So if you see this slide, for example, intelligence is a result of natural selection. So here is a group of individuals, okay, where we have primates. All are primates, right? Humans also can be classified as primates. Chimpanzees are our closest relatives, as you see in this phylogenetic tree. So there was an experiment done. If you go through this site, you will know about the experiment. A task was taught to this lower group of organisms and these little bit middle higher organisms and the highest evolved species on this earth, humans. So the task was forgotten by this lower group of organisms. They cannot repeat the same task for more than 1 or 2 times, whereas the middle group of organisms could repeat it 5, 6 times. And humans, we never forget, right? We do a work, we do something, and we remember for our lifetime, right? We don't excuse anyone for any mistakes done on us, right? So the idea here is the intelligence level is much more the brain size, right? So if you compare the brain size of these animals and these animals with humans, humans have a larger brain size compared with all this. That's why the intelligence level, the power to remember and replicate the same work done is much more higher in the humans. So when you say brain size versus body size, for example. So this is the horse, sheep, dog, and human brain, right? So you can see the human brains are much more bigger in size. 3 times more larger than it should have been among the primates also, because human brain is one of the organ that has been under intense selection pressure, because continuously we give work to our brain, right? So we don't continuously give work to our hands or to our legs. Sometimes we sit, sometimes we rest our hands, but even during sleep also the brain can work. Brain cannot sleep, right? So brain is under constant selection pressure, right, or evolutionary pressure. So that's why the size of the human brain is much bigger, right, than the other animals. And as we move into an agricultural society, into industrial society, into technological society, we give more and more and more work to the brain than to the physics. So even the future of the humans also, they predict that humans, the brain size is continuously increasing, and then it larger brain means more intelligence or more smart. So when we again talk about cultural and human evolution, right, our domestication of plants, we keep eating new varieties of food, right? So our system also needs to adapt to detoxify and digest and regulate those foods. We move on to colder climates, we move on to hotter climates, we keep traveling. So our body also has to, have some mechanism by which it can alter its own genes for better survival and adaptation under extreme conditions. Then, the process of fast food and cooking has led to lesser work to the teeth. We don't chew much food now like our ancestors, right? We have so much of pre-cooked or processed or ready-made foods, fast foods, that we give lesser work to the jawbones, like not like our ancestors. So our culture, our habit, our practices are also shaping the genes, right? Are also altering the genes. So here is one classical example of Tibetan people, right? So how do Tibetans thrive at high altitudes? High altitudes in the sense there is very low oxygen level. So how are they able to adapt, survive, and reproduce and stay happily generation after generations? Because for some of us, if we move to high altitudes, we will grasp for breath because there is low oxygen levels, right? So it is because of this DNA-based studies showed that there is mutation in the hypoxic gene, EGLN1, which is involved in the respiratory metabolism, right? And this helps in adaptation of the Tibetan population at high altitudes. So we are different due to climatic changes, due to different cultural changes, which keeps on shaping our body and our habit and habitats as well. So here is one slide which tells you about the mitochondrial gene. So mitochondrial genes we inherit only from the mother. It has 37 genes. So these are all the mitochondrial genes, ND1, ND2, then CO2, ATP6, CO3, ND4, ND5. So if we see from the same populations, right, a healthy normal individual, there is a present, whereas in the tumor patient, there is a G present. So there is a mutation from A to G for cancer in this gene. Even mitochondrial genes are also involved in cancer. Not only the nuclear 25,000 genes, we were talking about few genes, right? And it is specific only in these populations, this change. Similarly, the same gene, but at a different location, there's a change from healthy individual G to a cancer patient A. And it is found only in these populations. Similarly, in different Asian populations, in different genes, different mutations between healthy and cancer, in Korean populations, in Pakistani populations, are found. This is true for nuclear genes as well, right? So different individuals for the same disease have different genes involved. Not only different genes, even if the same genes are involved also, the mutations or variants can also be different. So population-associated diseases have to be studied. If we really want to solve any disease for any population, we need to look at the population first, and we need to understand the population. For example, at the disease level, the epidemiological or the demographic factors have to be studied, like lifestyle, food habits, ancestry, etc., etc. And for detection, diagnosis, and treatment, we need to study the genes, the genetic predisposition of the population for any particular disease. This can be also used for personalized medicine. And we know that more than 80% of the diseases that we are facing during the late stages of life are because of the genes and gene mutations or variants. So that is why the genomic backgrounds of the Indian populations needs to be studied first, along with the demography and epidemiological factors, if we really want to cure any human disease. So that is what today we call it as precision medicine, right, or personalized medicine. For example, we need to, for the proper medical care in the future, we need to have an individual's genomic data or gene data, their environmental data, environmental exposures, and lifestyle factors, whether they are smokers or non-smokers, right, alcoholic, any individual or a population, right, which will enable to treat the disease in a precise way. This slide. So DBT, New Delhi, the Department of Biotechnology, New Delhi, under Ministry of Science and Technology, have even started a project called as Genome India project, where the large focus is on medical genetics to identify variations in the genome that is disease causing, and also to determine how individual genes play a role in health and disease in an individual. So this type of DNA variations are common in normal human genomes, right? We saw that even though 99.9% of the genotype is the same, we have so many different ethnic backgrounds, cultures, right? Socio-cultural aspects, psychological aspects, disease aspects that we need to deal with. And it is also common in certain diseases like cancers and diabetes, etc., etc. And it is also showing in rare diseases like autism or schizophrenia or Rett syndrome. So the culture is humans structure themselves into cultures or ethno-linguistic groups, right? We always being, humans being social animals, we have a mechanism by which we have a tradition, we have a culture, we have a social system of learning, right? And we transmit it to our younger generations. We don't want our younger generations to deviate or divert into other cultures, right? So we have a social structure by which we have groups, right? Population groups. So hypothesis exists that the process of cultural evolution has played an active or a leading role in the selection of genes, right? Or even in the evolution of the genes. So many human genes that have been shown to be under recent or current selection are changing as a result of new environments. So humans also, we selectively transmit the variants that we have learned to our offsprings and to the others. So here is one slide which talks about gene evolution and cultural evolution. So gene evolution has been going on over time, and the cultural evolution also. But genes can, influence through development, right? It can influence the culture through developments, like modernized societies and things like that. And cultures have an impact. They modify the selection of the genes, right? So it is a two-way interaction. One interaction is it goes gene to gene, culture to culture, whereas the other is between gene and culture, right? There can be interactions. So it is genetic evolution and cultural evolution occurring parallelly. So there are various examples. Like one example is the lactase enzyme, LCT. So, like it has been found that in Western Eurasian and African dairying populations, means populations who were having cow, right, and doing dairy farming, this gene, right, which downregulates the lactase, the LCT gene, is rendered non-functional so that the adults can enjoy the milk drinking and they can benefit from the lactose. They can digest the lactase by rendering the gene non-functional. But in other populations, this gene has become functional in the sense people become lactose intolerant, right? They cannot take milk or they cannot take curd. So our in Indian population also, lactose tolerant milk drinkers in India, because we are the most largest milk producers and consumers also. So CCMB Hyderabad did this study and they found that Indian populations are also lactose tolerant. In the sense, this gene is rendered non-functional, like in Eurasian and African populations. So this is because of our habit of having dairy farming. Our LCT gene has been suppressed, and then we are still able to tolerate the milk. But certain individuals may have intolerance, right? So in a population, there will be always, confounders, and there can be always, some type of mismatches. But in general, biology and the culture are interlinked, and biological factors operate within a cultural context, and culture builds on biological foundation. So here is one another classical example of how sickle cell anemia came into practice, or came into the human populations. African individuals, right? They had a lot of malaria disease, and they were succumbing to malaria disease. But always there is a fight for survival, right? So human genes also fight for their own survival. So during that fight, it was for, what happened is the nuclei of our cells, from a round regular shape, they became a sickle shape. So that sickle shape, malarial parasites, the plasmodium, could not go and multiply in that. So Africans were becoming resistant to malaria by changing their round-shaped nuclear size to sickle shape. But later, the sickle shape could not carry proper oxygen across the body, and it became anemic disease. But even then, today also, we have survived malaria. The human race, I'm saying, survived malaria because of this acquiring of this anemic disease. So it's still okay, right? Instead of dying out of malaria, even though we are now harboring a additional disease, anemia, but we are surviving against malaria also, right? Now we don't die easily out of malaria because we are naturally resistant because of the sickle shape anemia prevalent in the human populations or human races. So this is one classical example of gene evolution. And another classical example is like Polynesian people. So there is a question: did a historical culture of exploration make Polynesian people more prone to obesity today? So actually, if we take the modern Polynesians, they have highest level of type 2 diabetes in their individuals. So people say that it might be because ancient Polynesians, they had a habit of exploring. So they used to travel across these type of seas without food and water for many days. So whenever they stop on a land shore, they eat lots so that for the next few days, they can survive without eating food. So that was their culture. But the modern Polynesians, they are not exploratory like their ancients. They have a sedentary lifestyle now, right? They sit and work in offices. So whether that metabolic activity that were being practiced by their ancestors, the gene responsible for, the sugar level. Basically, starvation and sugar levels are, like complementary to each other. Whether that sudden change of the lifestyle habits of sedentary lifestyle from a very active lifestyle to a sedentary lifestyle has resulted in gene mutations in them that can lead to diabetes, the metabolic alterations, right? Similarly, if you see the case of bottlenose dolphins, they forage, right? And then they have a spongy nose so that they can just at the root of the seafloor, they can look for their food. So whether this habit of searching for food, right, has altered their genetics, this cultural practice has altered their genetics to make a spongy nose for them, right, is one another point that is being well studied. So what makes us human? Genetics, culture, or both? So it is basically both, right? Some of these points we have already discussed, so I am not going to talk, but a few more examples. Now it is also possible to identify genes that may have specific role, the evolution of specific behavior also. For example, FOXP2 gene is involved in language, right? Acquisition or learning or use of the language. So modern individuals who have faulty copies of this gene have difficulty in speaking and understanding languages. FOXP2 gene is also the gene whose sequence has the most difference between chimps and humans, right? Even though chimps are our closest relatives. So the process of using the language, right? Learning the language and using the language, there is one gene that is evolving, right? Language is a socio-cultural aspect, right? Or basically a cultural aspect. So even there, a gene is. So Jim Davis, who has from Carleton University, is professor of cognitive science. He has spoken very nicely about this genes affect culture and culture affects gene. Like we saw the example of milk, lactose. He said that if a behavior is present in a human, it must have been encouraged by the genes or culture or both. Okay. So he gave various combinations. Genetic predisposition, right? If there is a gene responsible for a particular habit or a behavior, and if there is a cultural support in the society also, then the behavior will be definitely present. If there is genetic predisposition and there is no cultural support also, the behavior can be present because genes are entitled to bring in that behavior. Distinct. Genetic predisposition and there is a cultural inhibition, then the behavior may not be present or the behavior is in struggle, right? Hiding. If there is no genetic predisposition and if there is a cultural support, then also the behavior can be present because we go by societal rules and we abide by the societal laws, right? If there is no genetic predisposition and no cultural support, then the behavior is not present, then individual. No genetic predisposition and a cultural inhibition, the behavior is not present, right? Like, for example, let's take the example of drinking milk also, right? Or drinking alcohol, right? If you imagine drinking alcohol with this, the propositions of Jim Davis, you will better appreciate him. Yeah. If there is a genetic inhibition and the cultural support, the behavior may be present or it is in struggle. Genetic inhibition, no cultural support, behavior not present. Genetic inhibition, cultural inhibition, behavior. Right? The excellent theory put forward by him. He has worked lots about this gene, whether gene affects culture or culture affects the genes. So thank you very much for your patient listening, and I hope I'm on time and Professor Barthendu will not scold me that I have exceeded his time. But if I'll be happy to answer any questions, if there are any still. I hope the participants could take.
So you are very much in time, not
Okay.
exceeded. So there
Okay.
is one question in Q&A box, sir.
Sure. Q&A box. I think we have answered it.
Okay, already answered to us.
Answered it. Yeah.
So any other question, dear participants? Anyone?
Yeah, I hope the participants have some take-home message. I hope it was not boring for them. I hope there was some, because of interdisciplinary nature, right?
Yeah, yes.
Yeah.
So all thank yous and nice presentation messages are coming, sir.
Okay, thank you, doctor.
So thank you, sir, for sparing your precious time on this on Saturday, having a holiday for you. So
Holidays are a little bit better because we don't have any other academic commitments. Yeah.
and for a researcher, there is no holiday. Na? Even on Sundays, you are thinking of this line. Okay.
Exactly.
So thank you, sir, for joining us. And in future, also in some other programs, we'll again request you to be here. Thank you, sir. So on behalf of all the participants, on behalf of HRDC, on behalf of Professor Manoj, who is coordinator of this course, and I am co-coordinator of this course, on my personal behalf, I would like to say thanks to you for sparing your time and sharing a very good information with us. Because we are not from life sciences, I studied life sciences till class 10th only, biology. So it's good to listen these basic things from experts like you. Thank you, sir, for yes,
Thank you, doctor. Thank you for this opportunity, and it is very nice to meet the 25 participants, I guess.
Yes.
Yeah.
28 now.
28. No, including us, it is 28. Yeah.
Yes. Four